What if I told you that there's an important series of signals that happens within the neurons of our brain that controls critical cellular processes, but that we don't entirely understand how it works? The signaling pathway that I'm referring to is the mTOR pathway, and it is essential for regulating cellular proliferation, or the creation of new cells, cellular growth, which determines the size and shape of a cell, cellular metabolism, which is how a cell creates and uses energy to do basic cellular functions, and also cellular survival, which determines whether it lives or dies. In humans, a subset of epilepsy and autism spectrum disorders have been linked to improper signaling through this pathway during early brain development. My goal is to take a deep dive into the mechanisms of the mTOR pathway to allow us to better understand our brains in both health and disease. My work has helped to unveil a new model of adult onset epilepsy that will allow us to do just that. So under normal circumstances, a neuron will essentially survey its environment for signals like growth factors and respond through the mTOR pathway's cascade of events to control cell growth, for example. In the case of epilepsy, unusually increased pathway signaling during development leads to an abnormally hyperexcitable brain, presumably due to irregular neuronal growth and miswiring of important circuits. But beyond this general cause and effect, we still do not fully understand what is happening within a neuron or network once mTOR becomes overactive, like what genes are at play. Pinpointing these precise changes would make an enormous difference to our understanding of not only the development of epilepsy, but other mTOR-related disorders. So to help bridge the gaps in our understanding of mTOR, our model activates it by deleting a gene called P10 in the hippocampus of a mouse. The hippocampus is a brain region often associated with learning and memory, but it's also a region that we know seizures often originate from in humans. So here, P10 normally acts to stop mTOR from becoming active. So deleting it takes the brakes off and allows for increased pathway signaling in a subset of hippocampal neurons. By doing this, I can take otherwise mature neurons in the adult brain and trigger them to grow to more than double their original size, greatly increasing the number of connections they make with neurons they interact with. This leads to a more excitable brain that eventually produces seizures. So with our new model in hand, I will use cutting-edge molecular and functional techniques to help bridge the gaps between genes, neurons, and networks in hopes of finding a way to put the brakes back on in epilepsy. So at its most basic level, we understand that mTOR is overactive, but we do not understand the next steps down the line. We can therefore look at our problem as a series of basic questions. What is changing? Where are these changes happening within the hippocampus? When are these changes occurring as epilepsy develops? And how do these more specific changes directly influence the function of the brain? Luckily, with today's advances in gene profiling, we can identify new gene networks that are implicated in disease progression. First, ATAC sequencing takes us inside the nucleus, where we can identify regions of the DNA that lie waiting to be expressed as a result of mTOR activation. This will allow us to identify transcription factors that serve as network hubs or key regulators that function to turn on the expression of other target genes. Next, spatial transcriptomics brings us back out of the nucleus, giving us a hippocampus-wide view of what genes are actually being turned on or off within the networks. This will allow us to not only see how mTOR activation in our neuronal population affects that population itself, but also how those changes go on to influence the neighboring populations that they interact with. And by looking at these changes over time, we can pinpoint possible points where therapeutic intervention could be helpful in the development of epilepsy. Finally, I can take my newly discovered gene targets and directly manipulate them to understand how they alone affect function of the brain. By recording from the hippocampus in both mice and brain slices, we can see how our genes influence the way neurons now communicate with each other after the change. Like, do they become more excitable? Or even down to the level of how an individual neuron behaves within a now altered hippocampal network. So each step along the way takes us one step closer to understanding what truly lies beyond the mTOR pathway. In the end, I will identify new gene networks and their regulators that are directly responsible for forming and maintaining neuronal connectivity and function that become disrupted in epilepsy and other mTOR-related disorders. This exciting line of discovery will not only help us to better understand the human brain, but hopefully lead to the development of more targeted therapies in the future.